So this video is going to talk about the citric acid cycle or the Kreb cycle. And it is a cycle. It actually has so many different enzymes that work here, but I'm going to sort of whittle it down to the basics. So there's a lot going on that I'm not going to fill you in on just because I don't think you really need to know that. So what happens is, if a quick recap, we had everything taken outside the cell. So we had glycolysis get prepped, and it became 2 carbons and attached to acetyl-CoA. And now what happened is acetyl-CoA was able to bring those 2 carbons into the mitochondria, and now we're going to go ahead and do our next phase, which is called the Kreb cycle or the citric acid cycle. So what's going to happen is we're going to be able to move that 2 carbon molecule through a whole bunch of different enzymes, and they're going to slowly whittle off those 2 carbons until we have harvested a ton of energy. It's aerobic, it definitely needs energy, it occurs in the mitochondria, and it's going to do substrate level phosphorylation, which is another way of saying make ATP. So instead of reading you all these, you can actually read it on your own. I'm going to actually just explain it to you with a picture. So what happened is from the prep phase, we had these 2 carbon molecules that got attached to acetyl-CoA, and that made it through the citric acid cycle. You can call it the Kreb cycle, whatever you want to call it is fine. The coenzyme A detaches because basically it's just like a truck, and it basically delivered its payload, and now it's just going to go back to the prep phase and bring 2 more. What's going to happen is through these bunch of different steps, we're going to take that 2 carbon molecule and start to whittle it away. Through the process, we're going to make, for every rotation of this cycle, we're going to make, remember that NAD? It's that empty wheelbarrow. We're going to add some hydrogens and electrons to it, and then we're going to send it at the high energy form away. So this is kind of like the empty wheelbarrow, full wheelbarrow. In class I call this the uncharged battery and the charged battery, and it's going to go to our next step. We're able to take that adenine diphosphate and add a phosphate to it to become ATP, the higher energy form. So this can go somewhere in the cell, do some work, come back in this form, ADP plus a phosphate, and then a bunch of enzymes are going to stick them together to become ATP again, and that's what that substrate level phosphorylation is. Substrate level phosphorylation, it means just going to add a phosphate to it. And then we have a new wheelbarrow, a specific one. It's called FAD, and FAD is going to have 2 electrons and some hydrogens come to it as well. That's going to be the higher energy form is going to be the FADH. The lower energy form is called the FAD. So the FAD picks up the hydrogens and the electrons, and these guys are going to go to our next step as well. But if you look up here with the inputs and outputs, I told you it made 3 NADHs, 1 ATP, and an FADH, but you'll notice that the numbers are doubled. And the reason that is because remember how with 1 glucose molecule we make 2 acetyl-CoAs? Because that glucose molecule got split in half to become pyruvate. That pyruvate lost 1 carbon and got attached to acetyl-CoA. So both of them are coming in. So this is the first one, the second one's right on its way, and that's why these numbers are higher, because this is basically going to happen twice. So the acetyl coenzyme A is our input. We have 6 NADs, we have 2 FADs, and we have 2 of the lower energy form, ADP plus a phosphate. Because of this energy that's going to be harvested, we end up making a couple of things. We end up making 2 ATP, 2 FADH, and 6 NADHs. Also, every time we peel off one of these carbons, they're going to leave the body as CO2, and the only way we can get that oxygen is by breathing it in. So we breathe in the oxygen, it gets attached to this carbon, CO2 leaves. So that's why we produced 4 CO2s. So if we go ahead and look at it, what ends up happening is those two carbon molecules come in through these series of enzymatic reactions. One of the carbons are sort of whittled off, another one's whittled off. Through this process, we've gone ahead and energized 3 NADs to become 3 NADHs. We've made the FAD become FADH. And then we made one ADP plus a phosphate become ATP. And just remember, this happens twice because we have another one of these guys, these acetyl-CoAs, coming into the system to finish this off. So it'll go around once, and then the next one will come in, it goes around twice, and that's why we're still talking about how much energy we make with one glucose molecule. So, do we have a ton of energy right now? We kind of made a little, but we don't have enough to keep us alive. So far, we've made ATP, remember that's the currency from the cell. We've made 2 from the glycolysis, and look, I used the word net, and I used it because net basically is talking about, it cost us 2 to make 4, so this is the net number. We made 2 ATPs in the Kreb cycle. Remember, we didn't make anything in the prep stage. So we've made a grand total of 4 ATPs. That's not a lot, and if we only had 4 ATPs, we couldn't be higher-end organisms. We'd have to be like bacteria. So clearly that's not enough. So we've made a bunch of other high-energy molecules. We call them enzymes because they pick something up and deliver them. We've made, in glycolysis, we made 2 NADHs. In the prep phase, we made another 2 NADHs. In the Kreb phase, we made 6 NADHs and 2 FADs. So a total of 10 NADHs and 2 FADs per glucose molecule. These are going to go into our next step, and they're going to go ahead and make us a ton of energy. So our next video will talk about our next step, which is going to be called the electron transport chain.